Hey guys, welcome to Bio Tutorials with me, Emily. Just a quick note before we get started, I'm going to make and release unit videos throughout the year to help my sister, who's taking bio, and the rest of y'all in the same class survive the year and have a little bit of an easier time. So if you have any questions or anything you need a little extra help explaining, go to the TLC, and if you're still a little confused, then tweet me or comment, and I'll try to get back to you ASAP. Okay, let's get started. So today's topics are going to be Chapter 1, Introduction to Bio, and some other things you're just going to need to know along the way. Chapter 18, which is about ecology and populations. Chapter 19, communities and ecosystems. So just some key umbrella terms. Biology is simply the study of life, while ecology is the study of the relationships between organisms and their environment, so a little bit broader than just the study of life. Just as a piece of scientific background knowledge that you'll have to know from here on out is the difference between inductive and deductive reasoning. Inductive is less formal. It's known as discovery-driven science because a lot of times what they're doing is discovering something. There hasn't been much work on it before, so they can't really form a really good hypothesis based on any data that's been collected before. So what they do is they collect their own data, they do a bunch of studies, and then from all of that they funnel down and see, what did I learn? And they'll create a hypothesis or a theory from that. In deductive scientific studies, what they'll do is basically the scientific method. And they'll start with a hypothesis. Then they'll study what they hypothesize about, and they'll collect some data. And then they'll make a conclusion discussing whether their hypothesis was supported or denied by their results. And this sort of segues into the scientific method. Okay, so here's the scientific method, and it's here on a flow chart, and you're going to use this a lot this year in your labs. And what you're going to do is follow these steps. You're going to have a question, do some research, then you'll have your hypothesis, and you'll either be given an experiment or you'll create your own, and then you're going to have to analyze them, write a report, and draw some conclusions. Now there are a few things that are really important to remember when you're using the scientific method. First of all, you should only test one variable. So if you're testing what makes plants grow tallest, in each trial don't change the amount of water, the amount of sunlight, the type of fertilizer all at once, because then you won't know what caused your results. Secondly, you should use a control group. This is the group that will simulate normal conditions. So if you're testing which fertilizer works the best, you'll have a control group that just uses water, no fertilizer. Another really important thing to remember is to use more than one subject, because if you only use one subject and that subject is an anomaly, like a dead seed, your results won't be reflective of what would usually happen under the conditions you're testing. Along the same lines, use multiple trials, because one trial might not work as well as others, so you want to have more data. And finally, probably the most important thing is make sure what you're testing and how your experiment is set up really test what it's supposed to and the object of the lab. I find that people often have trouble discerning between independent and dependent variables. And so the difference is that the independent variable is basically what you're testing. So if you're testing how different plant fertilizers, say, impact growth rates of plants, the types of fertilizers would be the independent variable. So it's your choice. On the other hand, dependent variables depend on the independent variable. Because these variables are like the results. So if you are testing the impact of fertilizers on the growth rate of plants, the different heights of the plants would be your dependent variables. So they'd be different for each group, but they would depend on the independent variable or the type of fertilizer. Another broad thing you'll need to know before we jump into the nitty-gritty are the 3 domains of life, which are bacteria, archaea, and eukarya. Now, the main difference between these 3 domains are that eukarya have eukaryotic cells, so they have a nucleus, and these are generally more complex forms of life, like plants or humans. On the other hand, bacteria and archaea are prokaryotes, so they don't have a nucleus, and they tend to be less complex. Now we're going to get into ecosystems, which consist of all the abiotic factors, or non-living things, and the communities of species in an area. Those would be the biotic factors. Some examples of abiotic factors would be things like the water supply, the weather, or inanimate things like mud and rocks, while biotic factors would be things like this lovely bug or the plants in an area. Now, in a real ecosystem, the biotic factors like the bugs and the plants would form populations which would contribute to the community of species in the ecosystem. So what's the difference between a population and a community? Well, a population refers to only one of the species living in an area. So say this group of bugs would be one population because they're all the same species, or this patch of grass. It's all in the same area, it's one species. Now, what you could do is move these lovely bugs over here and refer to them both at the same time. So the grass and the bugs together would make a community because there are multiple species in the same area. But if you're only referring to one species, either the grass or the bugs, that would be a population. So an important branch of ecology is population ecology, and that's going to study things that influence a population's density and growth. A population density is the number of individuals in a certain amount of space, so area on land or volume in water. And to figure that out, you'll use a sampling technique. And that'll depend on whether an organism is sessile or not moving, so like a plant, or motile or moving, and that would be like a mouse or a living animal. So for sessile things, you would use a like plotting and counting technique, and what you do is you draw equally sized plots. Well, you'd plot them in real life, and just for these purposes, they're not really equal, but in real life they would be. And you'd count the number of organisms of that type in each of the plots. And you'd average that out. So say every square meter there were 20 of a certain plant, then you could figure out that there were probably 20 per square meter, and you can convert that to square mile or whatever, but that would be the population density. For motile creatures, what you do is the mark and recapture method. So you'd set a trap, and you'd catch a certain number of individuals, and then you would mark them with a tag or something, and you'd set them free. And then later you'd come back, set another trap, and you'd see how many marked ones you retrap. So say here I've trapped 8 individuals. I'd trap another 8. If the same 8 came back and were found in the trap again, chances are you're not going to trap the same 8 if there are other ones around. So you deduce that there are 8 living in that area. But if you deduce if you only find 4 in your traps, then chances are 8 was half the population, half of them were retrapped, and so there are 16 per that area. And you can find the density that way too. Patterns of dispersion is really important to population ecology because it can tell a lot about how organisms organize themselves or interact with each other. 3 major types are clumped, so that's the first one. This you could see in like schools of fish where there are groups of fish traveling together and then there might not be fish for a long time and then there'll be another huge group. Another type would be uniform. Those could be like penguin nests on an island or humans in the suburbs or in certain cities. And then random would be the last one. And there's no real rhyme or reason where things go. Those would be like trees in a forest where nobody really plants them. They just sprout up wherever they're seeded. We can model the change in the size of population over time with a population growth model. And one of these models is called the boom and bust. It looks like this. And it starts out with some exponential growth right along here. And then it drops off. This could be like mosquitoes, where during the summer, when the months start warming up, mosquito populations peak, and then comes winter, and they all die off. And then summer comes again, and then winter. And so the conditions are very favorable for a while, but then they become highly unfavorable, and the population dies off. Another model is called the exponential model, and this occurs under ideal conditions, and it looks kind of like this. You can see it keeps growing and growing, and it looks a lot like a J, so they call it a J curve, and that sort of is like the name for the logarithmic model, which will look kind of like this, and they call it an S-curve because it kind of looks like an elongated S. We're going to take a kind of closer look at this because unlike the exponential model, which might model bacteria or humans that have been growing in ideal conditions, the S-curve has a lot more factors in play that limit and encourage growth at certain times. So right here at number 1 is the lag phase. And the lag phase is where there isn't high population. See, there's not much here. And so it can't grow much. There might be a lot of resources, but with maybe only a few couples of organisms, they can't reproduce very quickly. At number 2, we have the exponential phase, and this is where things really start picking up because there's a lot of resources, there's enough couples to mate, and so population grows very quickly, like in ideal conditions in the J curve. Now, where this differs from the exponential model is at the top at number 3. Here, the population reaches carrying capacity. So the resources that keep the population alive is sort of in balance with the limiting factors. Here, there's no net gain or loss in population, so the population is dying as quickly as it's growing. There are 2 types of limiting factors that will keep a population from growing once it's reached carrying capacity. One of those types are density-dependent factors. Now, these factors will increase in intensity and severity as the population increases. These include things like a food shortage, a water source that is too crowded, or just not enough space to live. Density-independent factors, on the other hand, it doesn't matter if there are 2 individuals in the population or 2 million. The chances of this killing off an organism is equal regardless of how many are in the population. These include things like poor weather or natural disasters, and things like vector-borne illness. So whether you get bitten by an infected mosquito doesn't really depend on everybody that's standing around you. So we've talked a lot about individual populations, but now it's time to broaden it into the community, or a bunch of different populations of different species. Some of the defining characteristics of successful communities include, first of all, species diversity. For a community to be successful, there has to be a good give and take between different species. So they can't all be producers, they can't all be consumers. There has to be a balance. For a community to function, all of the species must find their own little niche. So they need to fill a space in the community that isn't already filled by something else. So, although there has to be a lot of different types of organisms for there to be this good give and take between the different species, there can only be so many different types because of concepts such as resource partitioning, where there's only so many resources and you can't have too many species competing for the same resources, and the competitive exclusion principle. This is sort of like resource partitioning in the way that organisms that are too similar can't live in the same community because they fill the same niche. For example, let's say green snake and pink snake both want to live in the same community, but they both eat the exact same diet of mice. Well, there are only so many mice, and so resource partitioning will make it very difficult for both species to live in the same community. And so eventually, one of the similar species, say pink snake, will outcompete green snake, and the pink snake will get the resources that they're both competing for, and pink snake will outcompete green snake and drive it out of the community. It'll die off. This is sort of survival in the fittest in action, because although pink snake and green snake were very similar, pink snake had a slight advantage that allowed it to remain in the community while green snake didn't. The prevalent vegetation in a community is extremely important because the vegetation is first what is at the bottom of the food chain. It feeds the rest of the community on some level, and it also defines basically what will live in the community. A community where tall grass is the prevalent vegetation will attract different animals than a community where, say, rainforest vegetation of shrubs and trees is very prevalent. Ooh, my thing seems to be skipping. Sorry about that. Okay, the third thing that a community really needs to be is stable. It has to be resilient to if one species becomes diseased, if there is a natural disaster that destroys some of the prevalent vegetation. It needs to be able to bounce back. And a well-constructed community will be able to do that because there is enough diversity and other vegetation that will allow it to do so. The final defining characteristic of a community that I'm going to talk about is the trophic structure, which is basically the way of saying who eats who eats what, who's the consumer, who's the producer, and at what levels do they feed, and we'll talk about that in a second. So, just a quick example, a very basic way of describing the trophic structure is snake eats Mickey Mouse. So at this point, the consumer is the or predator is the snake, and the prey is Mickey Mouse. Now, let's talk a little bit more about the trophic structure. So, I'm going to go over here, and we'll talk about food chains and food webs. So, food chain is just sort of a way of mapping out a line of consuming from the plant level to the as many. So first up in the food chain are the producers. These are the plants, and they get their energy directly from the sun, so they don't have to eat anything. They just fuel the rest of the food chain and produce the first level of food to fuel the rest of everybody else that will from then on eat. So the first species to eat, and the first level up from the producers, are the primary consumers. So I think first primary. These are species like chickens, and they are herbivores. Because they're primary consumers and only one level up from the producers, all they have to eat are plants, so they have to be herbivores. Next up are the secondary consumers. They are 2 levels up from the producers of the plants, so secondary. And they are species like humans that will eat the species like the chickens. Now, there are more choices when it comes to secondary consumers. They can be omnivores like the humans that eat both on the first level of producers or the second level of herbivores, or they can be purely carnivores. They'll only eat first order consumers. By the way, the order of a consumer, such as first order, second order, sort of corresponds with its name of primary consumer, secondary consumer. So primary, you'll think of first, so it's first order. Secondary has second in it, so it is a second order. Now, these names can go on and on forever, so primary, secondary, tertiary, etc., or first order, second order, third order. But eventually, these will have to die off and be decomposed. And this is the job of detrivores. And these are things like fungi, mold, bacteria, and they are what decompose the animals, like the secondary consumers. So when you die, these things like bacteria will decompose your corpse. What a lovely thought. Now, if that's not enough for you, let's talk about food webs. These are even more complex because they take into account that different animals might feed on multiple levels. So the first order consumers might feed multiple things. So the first order consumers, or primary consumers, will feed the secondary consumers. Also, when they die, if they are not consumed by secondary consumers, the detrivores will eat them. Or this could go another way. Secondary consumers might eat the herbivores, but they might also, like in the case of humans, eat the producers. And perhaps plant matter that is not eaten, like, say, a dead tree trunk or such, might also be consumed in a sort of quote-unquote way by the detrivores. Oops, sorry, gotta fix that arrow there. But you'll see that in reality, a community functions more like a food web than a food chain. The producers will feed different organisms. It just doesn't flow in one sort of direction. And there are consumers such as the humans, secondary consumers, that will consume on multiple levels. So they will consume first order. They will also consume producers. And the detrivores will, in the end, probably decompose just about any order of consumer in the map. Now, one more quick thing that's important to know about food chains before we move on is the transfer of energy down the food chain. As you go further and further down the food chain, farther away from producers, things get less energy efficient. Now, not pictured here is the sun. The sun is the most important source of energy on this planet, and it is what fuels the plants, which fuels the rest of the community. So the sun will transfer energy in the form of light to producers, which will transform that into plant tissues and into food molecules like starches and fats and proteins. So basically, these producers are very efficient in converting the sun's energy into consumable calories that can be transferred down the food chain. So 100% of the sun's energy on this level is transferred into food. So the food making is 100% here. This gets less efficient each time you transfer down. The amount of energy from the sun that turns into edible calories stored in each level, such as an animal like a chicken, becomes a lot less. So actually, it's only 10% of the previous level. So the previous level for primary consumers are the producers at 100%. So take 10% of 100%. 10% of the sun's energy is stored in a primary consumer as consumable calories. Now, why is that? Well, primary consumers eat the producers. And say they eat 10 pounds of plants in a day. 90% of those calories, so 9 pounds of those plants, will be gone to fueling their daily activities, their own life, pumping their heart, keeping them alive to roam around, find their own food. A lot of this will be burnt off in heat and such, and will be let out and wasted into other forms. Only 10%, or maybe one of the 10 pounds of plants, will be converted into the meat to build tissues that are consumable for the next order up of secondary consumers. So when a secondary consumer, say an omnivore like a human, decides to eat chicken instead of spinach, the amount of energy it takes to give them the calories, the same amount of calories, is 10 times as much when they eat chicken, because only 10% of the sun's energy is converted into consumable calories for secondary consumers, while, on the other hand, for a plant, it's 100%. So this is only 10% efficient, this is 100% efficient. Now, this becomes even worse when it comes to secondary consumers, because then 90% of, say they eat the primary consumer, the calories they get from that is for their daily life. So the calories you work off exercising, studying, going about your daily life, that is 90% of what you eat. 10% might go to building things like muscle. So at this stage, only 1% of the sun's energy is held in your consumable tissue. So as you go down, you can see the amount of energy that the sun gives to tissues that can be consumable by the next level drastically reduces. Only 10% is carried on from one level to the next. So this natural progression of losing energy, well, you're not really losing it, it's just sort of being wasted on natural processes of each order, is called the energy pyramid. And modeled like this, you can see that 100% of the energy the plants provide, but then it's only transferred to 10% of like consumable calories. 1 and then 0.1% of the sun's energy can be transferred into consumable calories. So it is highly favorable to eat on lower levels because you can get more of the sun's direct energy, you're more energy efficient, less energy has to be put into your diet to give you the calories you need. So we just discussed a bit more in depth about the trophic structure with food webs. Now we can discuss a little more about the stability of a community, which is another defining factor, in ecological succession, which is the community change when species replace each other. Now, stability has a lot more to do with secondary succession in being able to bounce back than primary succession, but we'll get to that in just a second. So, primary succession is starting from the bottom. You'll start with nothing. You'll have a pretty inhabitable environment, such as a new island where there's just rock, there's no soil, there's no place for things to take root. It's just a very plain rock or something like that, a desert, or nothing really exciting happening there. But eventually life will pop up and a pioneer species will take root. These will generally be very small plants because large plants take a lot more energy, nutrient space, and such, which the environment might not be able to provide. So small plants, very low maintenance, it helps a community take root. So once that species begins to thrive, a community will form. Now, these will consist of larger species because once the pioneer species have taken root, it will start to allow for a more habitable environment. Animals will come because plants attract animals, and the community will grow in complexity, and the species in it will become more complex, more large, etc. Now, secondary succession has a lot more to do with resilience in a community. It starts out, actually, with the community already. And what happens is the community will experience a disturbance. This could be something like a natural disaster, such as a wildfire. And although this might decimate all the populations living in this community, leaving no animals, no plants left, the soil will remain intact. Now, this is a lot better, the intact soil, than starting from nothing, because soil is already habitable. Things can grow from soil, as you probably can see from gardening or anything like that. So all this has to do is have pioneer species take root again, which will happen much quicker because seeds can just fall in and they'll start to grow again once the disturbance is over. And then after pioneer species, it will more quickly transfer to the community. A lot of the animals that used to live there will come back, and the community will thrive again. So it will sort of go in this cycle from, in the beginning, you'll start with nothing, then you'll have the pioneer species and the community. And from the community, A stable community with that good trait of stability will have this resilience to go through secondary succession. So when there's a disturbance, such as the wildfires that often happen in western United States on a pretty annual basis, the community will be able to bounce back. Even though there might be a disaster such as a wildfire, the soil will be able to sprout the pioneer species, the community will return, and then when another disturbance happens, the cycle will continue. Now, returning to that sort of theme of interspecial relationships are the different types of symbiotic relationships. So the three main symbiotic relationships we're going to learn about are parasitic, mutualistic, and commensal. And these names are assigned to different relationships based on the way that the symbiont, or the one that lives on or around the host, and the host species interact. And so in a parasitic relationship, the symbiont will benefit while the host will suffer. An example of a parasitic relationship is with things like parasites, such as tapeworms. If you get a tapeworm, it steals all your food and it lives while you become malnourished and might die. As the host, you won't be so happy, but the parasite will. In a mutualistic relationship, both the symbiont and the host will be benefited. So this is a pretty good deal. This type of relationship is a lot like flowers and honeybees. Now, the honeybees will get the nectar of a flower, and it will be able to make it into its honey and feed the other bees and such. And the flower benefits because its pollen is spread to other flowers, increasing its reproductive potential. A commensal relationship is one where the symbiont will benefit while the host really couldn't care less. It doesn't benefit or is hurt by the symbiont. They just live together. This might include the relationship between suckerfishes and certain types of sharks. The sharks aren't affected. The suckerfish doesn't hurt them or help them. But the suckerfish might be helped because scraps of the shark's food will float down and the suckerfish will be able to eat it. So it's easy access to food for the suckerfish, but the shark isn't helped or hurt. Okay, last thing, you're almost done, is the survivorship curve. The survivorship curve is very useful because it helps scientists look at the life history and plot it out of a certain species. And there are three types of survivorship curves that help classify the type of life history that a species might have. The first one is type 1, and it looks a lot like a sort of exponential decay model. And so what happens is at the beginning of life, surviving chances are very high. Not many die in the first few years of life, and as it approaches the max lifespan, quickly everybody dies off. This could be modeled with humans. Human babies, a lot of times, if they are born into a good situation, the babies won't die. We can provide babies with health care and such, so they will survive into adulthood. But as you start hitting the 60s, 70s, 80s, it takes a turn for the worse, and people start dying of old age and disease. They're more susceptible to illness and injury, so people are more weak, and they are starting to have failing health. They die. Now, a second form of life history is more linear. Basically, throughout its entire life, it has equal chances of dying from birth to death. This means that it could be like a squirrel, where in the beginning of life, the squirrel could fall out of a tree and die. And it could do that all through the rest of its life. It could get hit by a car. It could be whatever. And so there are risks throughout the lifetime which allow the rate at which, on average, the species dies is pretty consistent. Type 3 is basically the opposite of type 1. At the beginning of life, it is very risky to be alive, but once it passes a certain point, life is pretty good. You know, they don't really have to worry so much about dying off. This is like clams, where at the beginning of life, it's very risky to be alive because they are very vulnerable to other species that like to eat clams, like otters, where it's very easy to crack open a baby clam and find it and consume it. So a lot of the clams during this point of life die off because it's so easy to kill them. But after a certain point, it's harder to do that, and the rate at which they die tapers off. So once you hit a certain point, you're one of the survivors. You're less likely to die. So, just as a quick review, type 1, not as likely to die at the beginning of life, but as the species ages, it's more likely that they die. Type 2, very equal chance all throughout life, very consistent that they will die. And type 3, at the beginning of life, very high risk of death, but later in life, it's a lot more comfortable. That's all for today. If you guys need help with biomes, that was definitely not my favorite, but I could help you a little out with that. Please subscribe or follow me at Menagerie, and I can get back to you with any questions you might have with bio or chemistry if you know anyone who's taking it. So, thanks for tuning in.